other polysaccharide plants made out of cellulose, where every other glucose is in the opposite orientation, sort of upside down. Now, first you might say, well, big deal, it's still a bunch of glucoses all together. Does that really make a big difference? And it turns out, yes, it does make a big difference. This seemingly small change of flipping every other glucose upside down gives you a very different type of material. If you think about starch, like potatoes and bread, dough, and pasta, those aren't really tough materials. You can squish a potato with your hand, you can mold bread dough with your hands. It's not really tough in fibers, but something about this every other one upside down orientation in cellulose makes it a very tough, fibrous, strong material. So it's a lot tougher than starch, and it's inedible. We, members of the animal kingdom, don't have any enzymes that can digest this. And in a way, this underscores this point I made last week, that the active sites of enzymes are very specific for just the molecules they're supposed to be working with. And so that enzyme amylase that digests starch inside your digestive system cannot digest this, even though it's all made out of glucoses. They're just in slightly the wrong shape because everyone's upside down, and it just doesn't fit into your amylase, your starch digestion. So, all right, well, if it's inedible, what is it used for? Well, it's used for essentially building materials for the plant. The plant takes the cellulose that it makes and uses it to manufacture a cell wall. A cell wall is basically a tough shell that surrounds the cell. The outer part of the cell, well, I should say, the cell membrane is still there. It's this pink stuff made out of phospholipids. But phospholipids aren't very strong. They're not a good protective material. And so plants construct a cellulose cell wall outside of their cell membrane to make them more tough and more resilient. And since the entire plant body is made out of plant cells, therefore plant structures as a whole have a lot of cellulose in all those cell walls. And so that's why I was saying earlier, plant stems are very tough because they're all those celluloses. Wood is made out of mostly cellulose also. It's cellulose, but the plants make another molecule called lignin, which mixes in with the cellulose and makes it even tougher and harder. And that's why wood is more stronger and flexible than the cellulose you find in non-wood plant stems. The roots, the leaves are all very high in cellulose. Basically, all parts of the plant body that we see, like the roots and the stem and the leaves, are very high in cellulose. And even though we can't eat it, we use it for a lot of things. I guess I mentioned these earlier. Paper is made out of cellulose. Wood are made out of cellulose also. All right, so we actually know an animal has an enzyme that will digest this stuff. And I always thought it was kind of interesting because you think about, imagine a field of grass. I'm looking at the lawn out there. Well, it's mostly made out of cellulose, and so when you think about it, there's just as many glucose molecules here as there are in a big plate full of pasta. There's just as many glucoses there as in fettuccine or Alfredo or whatever, or rigatoni, whatever your favorite pasta is. And yet you can't eat it. It has no calories to it, but we can't digest any of those glucoses apart from each other. Now, some animals seem to be able to eat plants and get lots of good things out, like the cow munching on the grass out there. Well, the cow itself does not have any enzymes for digesting the cellulose that it eats. But inside the cow's rumen, I guess it is, inside the rumen stomachs, are some bacteria, and some bacteria do have an enzyme called cellulase that can digest cellulose. And so it's an example of mutualistic symbiosis. The cow gives these bacteria a safe, warm, moist place to live, and feeds them the cellulose. And in return, the bacteria essentially share some of the glucose that they get by digesting the cellulose with the cow. So both partners benefit from it. But it also underscores how tough cellulose is to digest. Even with these bacteria doing their best to digest it, the cow has to re-chew it. I don't know if you do this, but cows eat grass, and then after a few hours in their stomach, in their room, they throw it up again. And they chew it in their mouth and send it back down again to help the bacteria. And then a little while later, they'll throw it up a second time and chew it some more. I know it sounds kind of gross. And they'll keep chewing and re-swallowing and chewing and re-swallowing to assist the bacteria in digesting it. Eating some grass. Oh yeah. So we can't, even though it's full of glucoses, we can't digest them, so it has no nutrient value to us. But I was just thinking that now that they can move genes between species, why don't they take the gene from those bacteria that allow them to digest cellulose and put it inside human beings? And the advantage is you can eat grass and wood. It would be just like eating pasta as we are. And you might say, well, that doesn't sound very appetizing, and that's probably true. It would still probably really taste very good, but there's a lot of starvation on the planet, and so if we could allow people to eat grass and meat and stuff, that would just be more available for human beings. All right, so no members of the animal kingdom can digest cellulose, including insects. They are also members of the animal kingdom. But some creatures, like insects and shellfish, things like lobsters and crabs, have an exoskeleton which is made out of a cellulose-like material called chitin. What I mean by exoskeletons, insects and shellfish don't have any internal bones the way we do. But what they do have is a hard outer shell called their exoskeleton. And that exoskeleton material is made out of essentially something that's almost the exact same as cellulose. It's glucose with every chain with every other glucose upside down. But the only difference is instead of just plain old glucose, there's a modified form of glucose called glucosamine. And this is just a regular old standard glucose molecule. But instead of an OH down here, you have an NH in a amine group, and there's a couple other carbons and oxygen. Anyway, that's what glucosamine is. And you can actually buy glucosamine as a health food supplement. No, I don't. And it's supposed to be good for the joints. I don't know if it actually works or not, but some people take it to improve their joints. The polymer of it, the chitin, they found out makes pretty good stitches. I don't know how they found that out or where exactly they get it from, but probably from shellfish, like line them and turning into some sort of string-like thing. It makes excellent sutures. You might have heard of like some sets of sutures don't have to be removed if your body eventually digests them. Well, those are the chitin sutures that they don't have to take out because they dissolve themselves. I guess that's about it. Any carbohydrate question? Okay, well, let me tell you, I'll turn on the lights and tell you a little bit about the laboratory. The laboratory today is all about carbohydrates. And there's a few different activities, but one of the main activities is to analyze various foods, try to see which ones are high in starch and which ones are high in sugars, like sucrose. Towards the back of the room are various food samples. There's some bread and onion, and I think some potatoes, maybe an egg or something back there. And here's how you're going to be doing the analysis. Well, the handout will give you the exact directions, but this is the essence of it. You're going to order a pestle, and just like these alchemists back in the Middle Ages, you're going to just grind it out. You're going to get one food sample, some water to put inside there, and just grind it and grind it and grind it. your goal is to break apart, to push the cells up. And so if they do contain any starch and if they do contain any sugars, you're going to mash them so they spread out into the water. And then once you have mashed them up in your mortar and pestle, you're going to take some of the liquid, some of the watery liquid that might have that starch or might have that sugar, if there's any in there, and you're going to split it into 2 test tubes. Now I forget the exact volumes that we put in there, but I think it's 10 drops. I think you'll put 10 drops in this test tube and 10 drops in that test tube. Just follow whatever volume the directions say. In one test tube, you're going to test to see if starch is in there. And there's a special kind of iodine called Lugol's iodine that tests for starch. It's a pretty simple test. You basically just put a few drops of iodine in there. The iodine is in these light blue brown bottles. There's a reaction between this Lugol's iodine and starch. And it's like this. If there is starch, the Lugol's iodine turns a dark blue, or sometimes it's so dark it might look even black. If there is no starch, there is no color change, and you get the iodine color, which is kind of a yellowy color. It's certainly possible to have an in-between. If you get something that's kind of a light blue or a light gray, gray, which is light black, that there was some starch, but not a lot of starch. The other test you're going to do is called the Benedict's test. And it uses something called the Benedict's reagent, and it tests for sugars. I think it does test for all sugars. It's certainly monosaccharides, and I think that disaccharides is what it's called. And so notice that even though starches are